C3.2.7. Antigens trigger antibody production. Students should appreciate that most antigens are glycoproteins or other proteins, and that they are usually located on the outer surfaces of pathogens. Antigens on the surface of erythrocytes, red blood cells, may stimulate antibody production if transfused into a person with a different blood group. So antigens as recognition molecules that trigger antibody production. That's what we're going to learn about. The adaptive immune response is based on many specifics. So remember, there's the innate immune response and there's the adaptive immune response. Now we're talking about the adaptive one. Each type of B-lymphocyte makes a specific type of antibody. Each antibody is specific for one antigen. Each antigen is part of a specific group of molecules of a specific pathogen. Antigens are usually proteins, and many are glycoproteins. That's a bolded word, so be sure you write that down. Have it on a flashcard. Glycoproteins. Many are glycoproteins found embedded in the outer membrane of a pathogenic organism. This could be the plasma membrane of a bacterium or the outer cells of a protist or a fungus. Viruses do not have a plasma membrane, but they do have a protein coat called a capsid, and the capsid proteins act as antigens. An antigen, remember, is any substance that induces the immune system to produce antibodies. Most antigens are glycoproteins or other proteins, and they are usually located on the outer covering of pathogens. These molecules, rather than the entire pathogen, are the molecular antigens that result in an immune response. Okay, so your body doesn't recognize a bacteria. It recognizes a protein that is on the plasma membrane of the bacteria, and that protein is called an antigen. Other molecules besides those in pathogens can be recognized as antigens by our immune system. When organs are transplanted surgically, the organ or tissue transplanted must be matched very carefully by comparing the proteins of the donor and the recipient. So the donor is the one who gives the organ, recipient is the one who receives the organ. So they need to have very similar proteins on their cell membranes, plasma membranes. Transplanted hearts, kidneys, and skin are examples of organs that can be transplanted. Many proteins must be taken into consideration, and rarely is there a perfect match. The exception is when identical twins are used as both donor and recipient. So when one identical twin donates a kidney or a heart to their identical twin. Obviously, if they're donating their heart, it means they're dead. But a kidney, they could donate, or skin. Blood transfusions. That's when you lose too much blood and you need new blood, the hospital gives you blood from their blood bank. Okay, the blood transfusions should only occur after the blood types of both donor and recipient have been tested and are known to be compatible. Okay, so there are different kinds of blood types. You might know your blood type, right? And what that means, like what is a blood type? It means that you have a certain type of antigen on your blood cells. When a blood type is indicated by the notation AB positive, so if you're AB positive, that's your blood type, for example, this notation is actually providing information about two blood types. One is called the ABO blood type, and the other is the RH blood type. That RH stands for rhesus. In this example, AB represents the ABO type, and the plus, the little plus sign, the positive, represents the RH type or the rhesus type. So those are two different antigens. So there's the antigen that can come in the A or the B form, or the O form, which is not at all, okay? And the rhesus antigen, which can come in which either you have it or you don't, positive or negative. Blood typing is based on the presence or absence of three different antigens that are genetically inherited and found on the surface of erythrocytes. So these are proteins. There's three different kinds, three different proteins found on the surface of red blood cells. The three antigens are A protein, B protein, and RH protein or rhesus protein. Okay? The presence or absence of the three antigen proteins indicates a person's blood type. Okay, so there are two blood types, but there are three proteins. Okay? There's the A protein and the B protein. You can have one, the other, both, or neither. Okay? So if you have O type red blood, that means you don't have either. You don't have A or B. If you have AB blood, it means you have both. If you have A blood, it means you only have the A ones. Okay? And then the RH protein, you will either have it, which is positive, or negative, which means you don't have it. And here you can see this table where it describes the blood type, the name of the blood type. You can have blood type A, B, AB, or O, and what that means. A protein, B protein, A and B, neither A nor B. RH blood type, positive, negative. RH protein or no RH protein. A person with the blood type B negative has the B protein on their erythrocytes, but does not have either the A protein or the RH protein. A person with blood type O positive has neither the A nor the B protein, but does have the RH protein. For blood transfusions to be successful, a person must not receive a protein that they do not already have, as determined by their own genetics. A person with blood type AB positive can receive blood from anyone because they already have all three antigens. A person with blood type O negative can only receive blood from someone who also has blood type O negative because they have none of the three antigens on their erythrocytes. Okay. In a blood transfusion, a person cannot receive any of the three possible erythrocyte antigen proteins that they do not already have. This includes the A, B, and RH antigen proteins. Because what happens if you receive blood cells with antigens that you don't have? Your body will recognize those as foreign and think that those blood cells are pathogens. So the first blood transfusions were carried out before people had any knowledge of blood types. Transfusions were even carried out using farm animals as donors. As you can imagine, sometimes this accomplished more harm than good. Okay, so I just said that they recognize getting the wrong antigens will make your immune system recognize the foreign blood cells as foreign. Okay, we're going to read a little bit more about what happens with that. If someone receives a blood transfusion of an incompatible blood type, means a blood type with an antigen that they don't already have, a transfusion reaction will occur. The reaction is an immune response to what the body identifies as an antigen, an antigen on a pathogen. If someone receives type B blood and they do not genetically produce the B antigen protein themselves, antibodies will be produced that bind to the donated cells, and agglutination, which is clumping, can occur. Okay, agglutination is a bolded term. That's when you've got the antibodies they're shaped like a Y, and one sticks to one antigen and the other sticks to an antigen of another cell. You can get this kind of like clump forming. The resulting transfusion reaction may lead to minor effects, but it has been known to be fatal. C3.2.8, the role of helper T lymphocytes. Students should understand that there are antigen-specific B cells and helper T cells. B cells produce antibodies and become memory cells only when they have been activated. Activation requires both interaction with the specific antigen and contact with a helper T cell that has also become activated by the same type of antigen. So activation of B lymphocytes by helper T lymphocytes. Two important types of leukocytes that respond in the adaptive immune system are helper T lymphocytes, called T cells, and B lymphocytes, called B cells. Helper T lymphocytes chemically communicate with other leukocytes, including B lymphocytes, to signal the presence of a specific antigen. There are many types of helper T lymphocytes. Each type can only activate a specific B lymphocyte. The same antigen that activates a specific B lymphocyte will also activate a helper T lymphocyte. Helper T lymphocytes display antigens on their own plasma membrane. Helper T lymphocytes release molecules called cytokines. Remember cytokines from the earlier chapter? After finding a specific antigen to help activate a specific B lymphocyte. Some helper T lymphocytes are long-lived and are called memory cells. B lymphocytes produce a specific antibody that binds to a specific antigen. Okay, so B lymphocytes, they make antibodies. There are many types of B lymphocytes. Each type produces an antibody specific to one antigen. So one kind of B lymphocyte creates one antibody for one antigen. Each type that produces a specific antibody must be activated before it can make antibodies. Activation requires exposure to an antigen of the pathogen and also exposure to an activated T lymphocyte that is displaying the antigen and releasing chemicals called cytokines. Some B lymphocytes are long-lived and are called memory cells. So in C3.2 figure 1, we have activation of a B lymphocyte by a helper T lymphocyte. Both the B lymphocyte and the helper T lymphocyte have already encountered the pathogen. An antigen from the pathogen is displayed on the plasma membrane of the B lymphocyte and on the receptor of the helper T lymphocyte. A protein receptor on the B lymphocyte must match a receptor on the helper T lymphocyte. Cytokines from the helper T lymphocyte are released and taken in by the B lymphocyte. When all of these events have occurred, the B lymphocyte is activated. C3.2.9. Activation of a B lymphocyte results in cloning. There are relatively small numbers of B cells that respond to a specific antigen. To produce sufficient quantities, that means amounts, of antibody, activated B cells first divide by mitosis to produce large numbers of plasma B cells that are capable of producing the same type of antibody. The basis of the adaptive immune response is that a few specific lymphocytes of each type are present in the body at all times. So if you have, if you ever had the chicken pox, or if you ever had certain like influenza strains or something, your body has created specific leukocytes, specific lymphocytes here, and you still have them. A few of them are still left in your body. And once they get activated, because of you've been reinfected with it, then they start cloning like crazy. They start multiplying. When a specific cell type is needed for an immune response, activation of that cell type leads to cloning to make copies of that type of cell. So the main key point here is multiplication of activated B lymphocytes to form clones of antibody secreting plasma cells. This is so cool. I mean, it's complicated and messy, but it's so cool. The helper T lymphocytes and B lymphocytes described in section 3.2.8, the ones we just talked about, are antigen specific. Okay, that means that they only match up with one antigen, one pathogen. Antigen on a pathogen. The problem is that there is an incredible number of different antigens that may require a response. The immune system can only maintain a relatively low number of each type of cell that can respond to any one antigen. When specific B lymphocytes are needed in an immune response, they first become activated and then undergo numerous mitotic cell divisions. They clone. In effect, they create clones of cells that have the genetic instruction to synthesize mass quantities of the antibodies that can bind to the antigens of a pathogen. We have this amazing figure, okay? I'm going to walk you through this figure, okay? So, on the left is a representation of many types of B lymphocytes, okay, all different kinds. Only one type is activated by an antigen, and helper T lymphocyte not shown, obviously, because it also needs to be activated by that, but that part's not shown, okay? So only one type is activated by an antigen. The activated B lymphocyte undergoes repeated mitosis, okay? So you have all the different kinds of B cells in your blood, okay? Then the second column is showing activation. So an antigen binds to its specific matching antigen receptor, okay? So there's a leukocyte that matches that one antigen, okay? And then you have the proliferation into a clone of antibody-producing plasma cells, okay? So the activated B lymphocyte undergoes repeated mitosis to create a small army of the same type of B lymphocyte. A few of these, shown on the upper right, are memory cells and will not produce antibodies during the current infection. So a few are reserved for later on, for memory. The rest form antibody-secreting plasma cells. So that means they form these kind of cells that create the antibody and release it. Okay? That produce antibodies that can be useful in body tissues or be circulated in the bloodstream. Activated B lymphocytes become larger and develop mini ribosomes. Remember what a ribosome is? If you don't remember, pause the video and go look it up. They develop mini ribosomes, endoplasmic reticulum, and Golgi bodies. Ooh, all three organelles. If you don't remember, go review your organelles. All used for antibody production and secretion. So they create the antibodies and release them. Okay. What a cool figure. I'm just looking at it in the book. You're looking at it there, but it's really cool. In fact, you can even see the organelles there, right? The ribosomes, the Golgi apparatus, the endoplasmic reticulum. It's chock full of all that stuff. All right, C3.2.10, the role of memory cells. Okay, so remember, some of them don't go on to produce lots of antibodies. Some of them are put aside, kind of, to be memory cells. Students should understand that immunity is the ability to eliminate, that means get rid of, an infectious disease from the body. It is due to the long-term survival of lymphocytes that are capable of making the specific antibodies needed to fight the infection. These are memory cells. The adaptive immune response requires an initial, that means first, exposure to the antigen of a particular pathogen. Okay, so first you have to be exposed to the antigen or to the pathogen with the antigen on it. That first response is relatively long and is called the primary immune response. That's bold. Primary immune response. During this first exposure, there are no memory cells, as the few lymphocytes that can respond to this pathogen have not yet been activated or cloned. The length of time it takes for a primary immune response to occur varies depending on the pathogen, but there is almost always sufficient time for symptoms of the disease to develop. So you get sick the first time, or at least like a little bit sick. You start to have some symptoms. For example, it is the first time a person is exposed to a specific cold virus, they will have symptoms of the cold. The primary immune response is taking place while the person is experiencing the symptoms and will eventually result in the symptoms disappearing as the pathogen is eliminated from the body. So you get over the cold, right? Your body fights it off. The second or subsequent exposure to that same cold virus will trigger a secondary immune response. That's also bolded. Okay, so you've got primary immune response. We just talked about that. Now it's secondary immune response. The memory cells that were produced during the primary infection continue to circulate in the bloodstream. These very long-lived cells, now in relatively large numbers, are capable of responding to the same pathogen very quickly. It is usually so quick that symptoms of the disease do not present. It means you don't have any. Or they're quite minor. Sometimes we say mild. The secondary immune response not only occurs faster, it also produces many more antibodies than the first exposure. Okay, so we have a graph of this. We've got over time, and then on the y-axis you have antibody concentration in bloodstream. Okay, so the first point there is first exposure to antigen. That's when we're starting our time scale, right? And so you have the primary immune response, which you can see like some antibody concentration comes up in the bloodstream and then goes back down. So there's an initial response, but then if you get exposed to that antigen again, that pathogen comes in again, whoosh, your secondary immune response produces way more antibodies in less time. Antibody production by the primary and secondary immune responses. The second exposure to the same antigen may be months or years after the first exposure. The production of antibodies is quicker after a second infection, and the number of antibodies is greater. So you need to remember those two things there, that the production of antibodies is quicker after a second infection, and the number of antibodies is greater. We have no true immunity to a pathogen during the first infection, but we do have an immune system that is usually able to eliminate a new pathogen. True immunity begins with a subsequent infection as a result of the activity of memory cells. So the first time we fight it off, it's harder than the second time. C3.2.11, HIV transmission. Transmission of HIV in body fluids. Include examples of the means and implications of HIV, the human immunodeficiency virus, transmission. Okay. HIV, which is a boldest term, so you need to know about HIV, is the abbreviation, means the letters stand for something, for a virus called human immunodeficiency virus. Just like any virus, HIV is very specific about which organisms and which cell types in an organism it infects. Unfortunately, the host cells that it infects in humans is one of the key lymphocyte cell types involved in the human immune response. HIV does not survive outside the body and is not transmitted, that means by saliva, tears, or sweat. Okay, so you cannot catch HIV by touching someone's spit or their tears or their sweat. Okay. It is also not transmitted by insects, such as via mosquito bites. Okay, so you can't get it from a mosquito bite. The fluids that can transmit HIV are blood, semen, rectal fluids, vaginal fluids, and breast milk. Okay, so if you are exposed to those fluids, if you take those fluids into your body in some way, then you can be infected. The two most common ways that HIV is spread from person to person is by having unprotected sex with an infected person and by using a hypodermic needle, like an injection needle, that has previously been used by someone who is infected. So sharing needles, unprotected sex and sharing needles. Those are the main ways. Those are the most common ways that HIV is spread. In addition, it is possible for an HIV positive mother to infect her child during pregnancy, labor, delivery, or breastfeeding. So you can be born with HIV or you can get it from breastfeeding as a baby. In some countries, receiving a blood transfusion can spread HIV, but this is no longer a risk in countries where blood and blood products are routinely tested for contamination. So that's usually screened out now in many countries. Some medical treatments, such as injections for treating hemophilia, that's a genetic disease where you have trouble, where your blood has trouble clotting. Okay, some medical treatments, such as injections for treating hemophilia, have been known to spread HIV when the injected material was purified from human blood. In many areas of the world, these products are now produced by genetically engineered bacteria, and there is no risk of transmitting HIV. C3.2.12, the result of HIV infection. Infection of lymphocytes by HIV with AIDS as a consequence. Students should understand that only certain types of lymphocyte are infected and killed, but that a reduction in these lymphocytes limits the ability to produce antibodies and fight opportunistic infections. Okay, so it makes the body's ability to fight those first infections difficult. HIV is very specific about choosing which cell to infect. The host cells of HIV are known as helper T lymphocytes, or the CD4 T lymphocytes. CD4 is the name of the glycoproteins that are found on the plasma membrane of helper T lymphocytes and are used by HIV in its mechanism for entering a cell. Remember, it's a virus. It wants to get inside the cell. Unfortunately, for anyone infected with HIV, the helper T lymphocytes will eventually be killed by the virus, but they are the same cells used to activate B lymphocytes and some cells involved in an immune response. A person who has a very low helper T lymphocyte count in their bloodstream will not have a strong immune response to pathogens. This is the disease called AIDS, which is in bold, or acquired immune deficiency syndrome. Some people that are HIV positive and have progressed to AIDS are susceptible to opportunistic infections. So CD4 lymphocytes, AIDS, acquired immune deficiency syndrome, opportunistic infections. These are all terms you need to know. These are keywords that could show up on the exam that you need to be able to know to use in your answers. Get those points. These are infections that can occur more often or with more severity in people with weakened immune systems. Opportunistic infections include tuberculosis, salmonella, pneumonia, and several others. Deaths from HIV have greatly decreased in countries with advanced medical care where HIV-positive patients take daily doses of medicine that reduce the potential for damage to the immune system. Here we've got a map. The prevalence of people infected with HIV is unevenly distributed around the world. Many sub-Saharan African countries continue to show a high percentage of people infected. In this graphic, this map published in the peer-reviewed journal Nature, the data points are shown as much smaller, more accurate clusters than ever shown before. This can be used to help distribute resources or fight AIDS. Okay? So it shows the prevalence of HIV/AIDS in Africa, total percent of population, 15 to 49. C3.2.13, Antibiotics against bacterial infections. Antibiotics as chemicals that block processes occurring in bacteria but not in eukaryotic cells. Include reasons that antibiotics fail to control infection with viruses. Okay, so you can't take antibiotics to fight a cold. Bacteria are prokaryotic cells. Remember that? Humans and other animals are composed of eukaryotic cells. Go back to chapter A2.2 for a refresher. There are many structural and biochemical differences between prokaryotic and eukaryotic cells. For example, while protein synthesis occurs in both types of cells, the processes are different. Also, bacteria have a cell wall, a structure that is not characteristic of eukaryotic animal cells. Antibiotics are chemicals that take advantage of the differences between prokaryotic and eukaryotic cells. They selectively block some of the biochemical pathways needed by bacteria while having no effect on human or other animal cells. There are many categories of antibiotics depending on the biochemical pathway that is being targeted. One type of antibiotic selectively blocks protein synthesis in bacteria but has no effect on eukaryotic cells' ability to manufacture proteins. Another type of antibiotic inhibits the production of a new cell wall by bacteria, thus blocking their ability to grow and divide. Viruses have no metabolism, which explains why antibiotics have no effect on them. Viruses make use of our own body cells' metabolism to create new viruses. Any chemical that could inhibit viral metabolic activity would also be damaging to our own body cells. Antibiotics should not be routinely prescribed for viral diseases. Okay, so that explanation there is really important to know. There are chemicals that have been developed as antiviral medications. These chemicals suppress a virus's ability to infect and multiply in the host's cells. Antiviral medications have become the standard of care for people with HIV and hepatitis C infections. C3.2.14, Pathogenic resistance to antibiotics. Evolution of resistance to several antibiotics in strains of pathogenic bacteria. Students should understand that careful use of antibiotics is necessary to slow the emergence of multi-resistant bacteria. Students should recognize that the development of new techniques can lead to new avenues of research. For example, the recent technique of searching chemical libraries is yielding new antibiotics. Bacterial resistance to antibiotics is a serious problem around the world. For too long, antibiotics have been used improperly and too frequently. The fundamental principles of evolution explain how pathogenic bacteria become resistant to any one antibiotic. When bacteria find their way into living tissues, frequently the environment is nearly perfect for their growth and cell division. Tissues are moist, full of nutrients, and relatively warm. In this type of growing environment, some pathogenic bacteria can grow exponentially and double their numbers in as small a time period as 20 minutes. Every cell division requires the DNA of a bacterial cell to replicate. Mutations occur spontaneously when DNA replicates. Most of the mutations are of no consequence, but when the DNA replication rate is very high, one or more mutations is likely to occur that is consequential. One of those random but consequential mutations may give a bacterial cell protection from the biochemical action of a particular antibiotic. One mutation is all that is required, as that bacterial cell will undergo binary fission repeatedly to grow into many cells. All bacterial cells that arise from the mutated cell will have the resistance to the antibiotic. In addition, the mutated strain may now cause infections in other people. Okay, let's look at this picture. Bacterial species grown in a petri dish showing resistance to antibiotics. So that dull, cloudy area, that yellow, cloudy stuff, is where bacteria are growing. The small white discs are impregnated with antibiotics. So those are little pieces of paper with antibiotics on them. The clear area around many of the discs indicates that the antibiotics are preventing bacterial growth. The clear area is called a zone of inhibition, bolded term. The presence of bacterial growth around 3 of the discs indicates those antibiotics that have little to no ability to prevent growth. Okay, so you see the 3 that don't have any zones of inhibition around them? It means they weren't any good at stopping this bacteria. A great deal of scientific research precedes the release of a new antibiotic before it can be prescribed. Antibiotics always come with directions for use that are based on this research. These directions include how long you should take the medicine for. If someone stops taking an antibiotic early, for example because their symptoms have improved, only the bacteria that are most sensitive to the antibiotic have been killed. A few resistant cells may not have been killed and can grow into a new resistant strain. You should always take an antibiotic for the full prescribed duration. That means if you get antibiotics for a urinary tract infection and you're supposed to take those antibiotics for 3 weeks, you should take them for 3 weeks even if you no longer have symptoms after 1 week. A few strains of pathogenic bacteria have emerged over the years that are resistant to several antibiotics. One example is methicillin-resistant MRSA, Staphylococcus aureus. MRSA. This bacterial strain causes staphylococcal infections. And that's a bolded term. You're going to need to practice that one a lot if you're going to spell it right. I would need to. Staphylococcal infections. Staphylococcal. Staphyloccoccal. Staphylococcal. Staphylococcal infections that are very difficult to treat. Responsible use of existing antibiotics is needed to prevent the emergence of more multi-resistant bacteria. Responsible uses include only prescribing an antibiotic when it's necessary. Don't pass them out like candy. Taking the full course of an antibiotic and not stopping when symptoms first subside. Reducing the spread of bacterial diseases by vaccination, hand washing, and proper food hygiene. Reducing or stopping the practice of adding antibiotics to farm animal feed. Okay. Sometimes when animals are raised in close conditions, disease can spread really fast. Bacterial infections anyway, so they just add antibiotics into their food to kind of prevent that from happening. The development of new techniques can lead to new lines of research. The use of chemical libraries is a new approach to antibiotic development. Chemical libraries store chemicals along with all the known information about those chemicals. Searching chemical libraries is yielding new antibiotic treatments because synergistic effects of antibiotic combinations can be explored at the molecular level. Methods using artificial intelligence are being used to screen for new antibiotics. This involves predicting the interaction of chemicals using AI and the known chemicals in chemical libraries. Halicin, a chemical screened by AI, is a new and promising antibiotic that is being prepared for clinical trials.